Hello and welcome to this Nature Research custom media webcast titled Intracellular Drug Concentrations Achieving Accurate Concentration for Novel Drug Modalities. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Chemica. While the cell is the basic unit of life, the metabolic and disease-associated pathways that drug developers aim to modulate are at the subcellular level. As a result, conventional methods for evaluating drug exposures, which rely on cellular measures, can fall short. The principal readout in the field of pharmacokinetics is the concentration of drugs in biological matrices, but the typical format of these matrices, biofluids, tissue, and cell lysates, isn't sufficient to further our understanding of drug biochemistry at the level of the single cell or organelle. This webcast will describe a recently developed and validated mass spectrometry imaging approach to gain access to this final frontier of pharmaceutical bioanalysis. We'll begin by hearing from Dr. Michael Kurczy, Associate Principal Scientist at AstraZeneca. Mike will discuss how drug discoverers can ensure their molecule reaches the desired target and completes its therapeutic mission, how drug exposure can be accurately measured at intracellular and organelle levels, and what database tools are available to provide continuity from experiment to experiment. We'll then move on to a question and answer session with you, the audience. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them at the end of the session today. And now, over to Mike.
Hi, my name is Mike Kurczy, and I'm an Associate Principal Scientist at AstraZeneca, located at the site in Gothenburg, Sweden. And the title of this webinar is Intracellular Drug Concentration, Achieving Accurate Concentration for Novel Drug Modalities. And so our approach to this uses mass spectrometry imaging. And specifically, we're using NanoSIMS imaging, which is a technique that I would say most people working in pharma don't have a lot of experience with. So in addition to showing you how we do this, I also want to kind of demystify the technology and really make the case that this technology is actually very accessible to the field and that it has the potential to answer some of the fundamental questions that seem to keep popping up in this area. But first, I'd like to talk about how NanoSIMS ended up at AstraZeneca. So around 2015, Professor Andrew Ewing from Gothenburg University secured funding for the first NanoSIMS in the Nordic countries, and this is from the Wallenberg Foundation. And so Andy, with support from the local universities, including the Chalmers University of Technology, formed the Chemical Imaging Infrastructure, so the CII, to manage the NanoSIMS along with some of the other mass spec imaging instruments in the area. And so, recognizing the collaborative opportunity, they took the decision to install the NanoSIMS in the AstraZeneca BioVenture Hub. And so the BioVenture Hub is this kind of framework that allows emerging life science companies and academic groups to operate in AstraZeneca with a kind of open innovation ethic, which is supposed to generate innovative ideas like using a NanoSIMS to measure subcellular pharmacokinetics. And so the environment is very exciting. And there's always a lot of ideas coming from both the academic side and the AstraZeneca side. But this collaboration kind of coalesced around the problem presented by new drug modalities. And very, very simply said, these molecules, these new drug modalities, they don't reach their targets the way that small molecules do. They rely on a physiological process that happens at the subcellular level. And so I'm talking about endocytosis. And this cartoon shows that when the drug is endocytosed into the cell, it's still separated from it by this endosomal membrane. And so in other words, even when it's in, it's not really in. And so to be productive, the drug needs to find its way into the cytosol. And so ultimately, what this means is that the concentration of the drug that the cell is being exposed to is not the concentration in the plasma or even at the tissue level. The interface where the drug exposure occurs is at the endosomal membrane. So what we wanted to do was quantify this physiological process, this endocytosis of these drugs, and relate that to the effect, which we thought we could do with the NanoSIMS, as we like to think of what we do as subcellular functional imaging. And so in the spirit of kind of demystifying the NanoSIMS, I want to compare it to something that I think most people would be familiar with. And that's functional imaging with positron emission tomography. And so in this example, if you're interested in studying Parkinson's disease, you might want to measure the capacity of dopaminergic neurons to store dopamine. And you would do this by introducing a radio-labeled molecule called L-DOPA, which is the precursor for dopamine and also a treatment for Parkinson's disease. And you would administer the radio-labeled L-DOPA to the patient, and if the neurons were storing dopamine like they should be, then you would see an image that looks like this. So you'd see a kind of bright emission coming from the region where there's a high density of dopaminergic neurons. But if we could zoom in on this region, this is supposed to be a dopamine neuron, you would see that the signal is not coming from the cell itself, but it's actually emitting from the secretory vesicle where the cell stores the dopamine. And you might also notice that the L-DOPA is not L-DOPA anymore because it's been converted to dopamine by this enzyme, this decarboxylase. And so this was very interesting to me because L-DOPA is a drug and a tracer, which is analogous to what we want to do. We want to trace our new drug modalities. And it's also analogous since the dopamine is stored in these secretory vesicles, which to me look a lot like the endosomes that we're interested in. So as a proof of concept, we decided to use an L-DOPA molecule that had been modified to be a SIMS tracer. So we could represent this process, kind of as it's seen in this cartoon. And so we did this in vitro. And this is a TEM image of the cell that we used. And it's a rat pheochromocytoma cell, which expresses both the enzyme and the transporter. And the secretory vesicles that we're looking for are actually quite easy to see because they contain these dense cores. You can see one here, here. And so they show up really well. And so if the sensitivity of our tracer is right, and the cell is storing the dopamine the way we expect it to, we should see our SIMS tracer located in these spots, co-localized with these spots. And we were very happy to see that it worked out really well. So I think the best way to look at this data is to maybe pick your favorite constellation in the TEM image and then see what it looks like in the SIMS image. And if that's too hard, we did a zoom in and an overlay, and you can see this nice set of 5 here next to the nucleus. But this was really encouraging. So we had a drug that was a tracer that we could measure in a subcellular structure. But this is not endocytosis. And in fact, the transporter that loads these vesicles is extremely efficient. And so we wanted to find a more appropriate model. And luckily, there's another PET tracer that we could use for inspiration. And this is GLP-1, which is a peptide, which is specific for beta cells because the beta cells express the G protein coupled receptor GLP-1R. So you radio label the GLP-1, and it's recognized by the receptor and pulled into the endosomal space where it accumulates for a little while at least, and the signal coming from the endosome is what allows you to find the beta cells. So as another proof of concept, we did another in vitro model, and again we took TEMs before we did the SIMS imaging. So this is a HEC293 cell overexpressing that GLP-1 receptor. And this time we're looking for the endosomes, which are a little different than the secretory vesicles. They're not quite so uniform in size, and they don't have that dense core, which makes them easy to find, but they're still fairly easy to find, these clear structures. And so we had the same perspective here. If the sensitivity was good enough and the receptor was doing what we had anticipated it would do, we would see an accumulation of our tracer in these endosomes. And again, we were very happy to see that it worked out really well. And this was really, really exciting because now we had a drug. GLP-1 is the basis for a few drugs that could be used as a tracer and was taken into the cell via endocytosis, like the new modality drugs that we wanted to study. So it was really just a matter of turning our drugs into tracers at this point. So turning our drugs into tracers. So most of our work has been done using antisense oligonucleotides, so ASOs or ASOs, which is a single-stranded nucleotide. You can see that in the cartoon. And we have a lot of great oligochemists at AstraZeneca, but I want to take the opportunity to kind of demystify the labeling strategies for NanoSIMS. So when the chemist makes our tracers, they aren't functionalizing some exotic label to the molecule. They're synthesizing the molecule using labeled starting materials, which to me at least seems like a much more straightforward approach. Another advantage is that the labeled starting materials that they use, they're not radioactive, which makes it much easier to convince them to use in their synthesis. So we've had most luck with halogenated bases. So if you can find a spot for an iodocytidine or a bromothymidine, you're ready to go. But the Astra chemists also developed a really elegant strategy where they replaced the sulfur on these PS linkers, which is a pretty common linker, with sulfur 34, which allows us to see this. And this is a really convenient synthesis because you don't need to consider the sequence when you make the tracer. But these tracers are cold, meaning that they don't emit. So to get them to emit, we need to use the NanoSIMS. And so next part is the NanoSIMS. So the NanoSIMS, we start with the name, nano, that part is easy, 10 to the minus 9 as in nanometer, and SIMS stands for secondary ion mass spectrometry. So to measure secondary ions, you need to start with a primary ion. So we use a primary ion beam, which we focus really small down to 50 nanometers, and we raster that across the sample. And so maybe you noticed that in the examples before, we show that we always have an EM, electron microscopy image. And that's because our samples we use are really thin, really flat electron microscopy images. So this samples rather. And so this would be an EM section down here. So the primary ion beam impinges on that surface, and all that energy produces ions, secondary ions. And so we send those secondary ions to the mass spectrometer, where we measure 7 masses. including our tracers, so our iodine, our bromine, and our sulfur. And since we're keeping track of where this primary ion beam is going, we can generate an image at each channel. And so here we have an image of a human hepatic spheroid, which is a very cool model, 3D cell culture. And we treated that with an iodine-labeled oligonucleotide and a bromine-labeled oligonucleotide. And so we also measure this carbon and nitrogen ion, which gives us an idea of the cell structure. So I think you can see that there's a nucleus here and a nucleus here. And so I mentioned earlier that we analyze electron microscopy sections. So it would be strange if we didn't do electron microscopy on the sample before we took the SIMS. And so we do. And I think if you compare this to the CN image, I think you can tell that these are an image of the same thing. And so we use the common landmarks on these two images to kind of align the EM, and then that allows us to add that EM level of spatial resolution to the SIMS. And the only thing I'll say about this experiment is that these oligos are identical except for the labeling. So there shouldn't be any difference in the uptake or the trafficking, but we see them in different places. We see the iodine by itself, or not by itself, but with the bromine, and the bromine here by themselves. And so the only difference between the two is that we treated the iodine oligo with a couple hour delay. So really what we're seeing is that this endosomal structure is a few hours younger than the ones that contain just the bromine. So now that we're all SIMS experts, we can move to some more applications. So if you remember a few slides back, the GLP-1 peptide is a really good way to find beta cells when you're using PET imaging. And my colleagues at AstraZeneca realized that this peptide might also be a good way for an oligonucleotide to find a beta cell. And so conceptually, it's similar to the PET experiment. Instead of using a radiolabeled peptide, you conjugate your oligonucleotide to the peptide, so that you can target the beta cell. So the receptor kind of works like a homing beacon for the drug. And so the peptide finds the receptor, and it gets pulled into the endosomal space, just like the PET experiment. But for the oligo to work, at least some of it needs to get out of that endosome, and it may need to decouple from the peptide. And so related to this, my colleagues made an observation that the stability of the linker that connects the peptide to the oligonucleotide was related to the effect they saw in vivo. So we took two of the more interesting ones. So here is a malamide linker, which was less stable but effective. And this is the quick linker, which is a bit more stable and wasn't quite as effective. And the chemist graciously labeled the peptide part with iodine and the oligonucleotide part with sulfur 34. And so the hypothesis was that the instability of this malamide linker allowed more of the oligo to escape the endosome, which increased the efficacy with respect to the more stable click linker. So the test of the hypothesis was to measure these dual-labeled conjugates in the endosomal space and compare the relative tracer signal from the peptide to the oligonucleotide. And we did this again in vitro, and we used our trusty HEC293 cell overexpressing GLP-1. And the top row is the malamide linker, the less stable, more potent. And on the bottom is the less potent click linker. So in the first column, we have the CN image, which gives us an idea of the structure of the cell. And here is the nucleus, and here's the cytosol, where you expect to see the endosomes. And down here is here's the nucleus and the cytosols in these regions. So if we go to the last column, which measures the iodine, we see, which measures the iodine, which is tracing the peptide. We see that the receptor has done what we expected it to do. It's grabbed onto the peptide and pulled it into the endosomal space. And you can see these bright spots here. But if we look at the sulfur 34, which is tracing the oligonucleotide, we see very little signal for the weak linker and a little bit more for the stronger linker. So we had some evidence that the linker chemistry plays a role in the escape. But from an analytical perspective, it's still relative quantification. And at the beginning, I said we want to report exposure, which needs to have a unit like moles per liter. So it was really important for us to figure out how to report a concentration. So to figure out how to do this, we took a step back and we took a really a material science approach and tried to define the sample as a bulk material. So we look at our sample as a material that contains biological inclusions. And since I'm talking about sample prep, I want to take the opportunity to demystify this part as well. When we have our samples prepared, what we have done is a standard electron microscopy type sample prep. And there's lots of labs that have this expertise. And we work with a really great one, the Center for Cellular Imaging, which is in the core facilities at the Gothenburg University. And it works really well for us to send them our fixed cells or tissue, and they process the material, and then they send the samples back to us mounted on the substrates that we use. So just briefly, we take our treated sample and chemically fix it. And the sample is dehydrated, so all the water is removed and replaced with this embedding epoxy. The epoxy gets cured, and then they trim it to this pyramid shape, and they cut some sections, which are quite thin. It says less than 300 nanometers for TEM, but if you use SEM, you can go up to 500 nanometers or even a micron. And this cartoon shows what we end up. It's a resin material that includes our drug, that's the green part, and the cell material. And so to figure out how to quantify, we decided to break the sample down into three components. So the first part is the epoxy resin, which is most of the sample, and especially considering that the cell is also 70% water. And the next part is everything in the cell that's not the drug, and we call that the biomass. So if we're using a labeled drug, and here we're using carbon-13, the idea was that the carbon from the epoxy and the biomass was basically diluting the signal coming from the drug. So if we could figure out the concentration of the carbon in the epoxy and the biomaterial, this would allow us to translate our signal, SIM signal, into concentration. And so we worked this out using the dopamine system that I talked about a little earlier. And this was actually the perfect system to do this, is because there's actually an orthogonal way to measure the number of molecules in the vesicles in situ using electrochemistry, and this is an example of an idea that came from the university side at Professor Ewing's lab. And what we showed was that the SIMS measurement agreed very well with the electrochemistry measurement, and they were totally independent measurements. So the big take home was that under the right conditions, the SIMS sample was a good reflection of the cell in situ, including the concentration of dopamine in the vesicle. And we further found out that the embedding resin was well matched in terms of carbon content to the cell. And so this opened up the possibility for us to generate calibration standards. And so we call this our Rosetta Stone experiment. And so this led to what we call the SIMS workflow. And the workflow really hinges on that sample preparation, which includes that resin embedding step. And as I mentioned earlier, the resin is actually very well matched to the carbon in the biomass. So we use that as you would blank plasma to generate calibration standards. So we take our tracers and we mix them in with the epoxy. We cure it like we would with the sample. We section it and analyze it exactly the way we do with the sample. And then we generate a calibration curve. And then that calibration curve allows us to translate these SIMS images into concentration images. And I will say that our standards are freely available to anyone interested in doing a quantitative experiment. And we really encourage you to reach out to us. And so the physical part of the drug SIMS workflow is really just those standards, but the spirit of it is the validation, which is a longer story. But I will show this one experiment where we treated human hepatic spheroids again with 3 different types of antisense oligonucleotides, or 3 different labels. The oligos were identical except for the labeling strategies. And we found that the average concentrations were quite repeatable across the different labeling strategies. And so each one of these dots is like an ROI that you see in this image here. And so these were all incubated at 5 micromolar. And you can see that the endosomal concentration is about 200 micromolar. So this is like a 50 times concentration differential. And so we were very interested in this, and we wanted to look at this a little deeper. So we designed an experiment to look at the relationship between the incubation concentration and the intracellular concentration that we measure using the drug SIMS workflow. We chose 3 concentrations, 100 nanomolar, 1 micromolar, and 10 micromolar. And so this is kind of a truncated version of an in vitro screen that you would do to look at the potency of these molecules. And so what you would do is you would use these concentrations as reference points. And so we use the hepatic spheroids again. And this is in the top row, you have the CN image, which kind of gives you the structure of the cells. And there's several cells in each image. And when we look at the sulfur signal, which is tracing the oligonucleotide, we see that we do see more oligo when we have a higher concentration, and that's not so unexpected. But if we look at the individual measurements, so each one of these dots is an ROI, we see that the average concentration doesn't increase tenfold like the incubation concentration does. And this is easier to see when we plot the intracellular concentration versus the incubation concentration. And so the point is that if you do use the incubation concentration to say something about potency, you're convoluting potency and the uptake and also the endosomal escape all in one measurement. And so basically we're saying that being able to measure the exposure closer to the site of action could really help shed a lot of light on this this very tricky and interesting problem. And so just to conclude, I hope you've seen that it's possible to measure oligos at the subcellular level, and that the sample prep and labeling strategies are accessible to researchers working with this kind of drug modality. The intracellular concentration does appear to be related to activity. As we increase the incubation concentration, which increases the effect, we also see an increase in the intracellular concentration. So that's not surprising, but we also observe that it's not proportional. So in vitro experiments, at least, where we judge potency from the incubation concentration, we miss this intracellular aspect and convolute potency and uptake. And I think the same principle will apply in vivo to a greater extent, where the variety of cells will further convolute the exposure at the target cell. And for this reason, we think that absolute quantification of oligos closer to that site of action in the endosomal space will be a valuable addition to the pharmacokinetic studies that we do for oligonucleotides. So I have some acknowledgements. So I mentioned most of the key players here on the collaboration slide, but I didn't mention SSF, who's funding Cecile Beckhart's PhD, and she's been a real big part of the success of this program, and we're really happy to have had the chance to work with her. I also like to mention Kamika, especially Aurelian and Marion, who immediately recognized the value in what we're doing and agreed to help us get the word out, because ultimately what we want is to have more people engaging in this technology so that we can build an intuition around what these drugs are doing at the subcellular level. And finally, I thank you for your attention.
Thank you very much for that presentation. It is now time for the live question and answer session. To ask a question, please type it in where it says type your questions here, and then press submit. We do have a good amount of time for questions, so do please feel free. Okay, Mike, to get us started then, I would like to ask you this, or to sort of summarize. It seems like the advantage of new drug modalities is that they simplify the drug discovery process, since the drugs are not as diverse compared to all the possible small molecule drugs. So on the surface, it seems like the oligos are like one molecule, and that just by changing the sequence, you could treat different diseases. Do you envision that there will be a general predictive model for these molecules that could be used to circumvent the need for pharmacokinetic measurements altogether?
Yeah, that's a good question. I mean, first, I would say maybe simplify isn't exactly right. You sort of get rid of all the small molecule problems, the screening of millions of molecules, and to optimize for potency. But you add these sort of larger molecule problems, which we really need to we need to figure out if we are going to be able to target any disease with this kind of drug. I don't know. I mean, my feeling about this is that, I mean, the more we look into what happens at the subcellular level, and the more we figure out the basic principles of these things. I mean, it's biophysics and it's biology, obviously, but I think that there is a possibility that it could come to a level where we have enough data where, I mean, I guess ideally this is where you would ask ChatGPT, how do I cure my diabetes? And it says, okay, it looks like so I think that's kind of like the dream, but yeah, there's a lot of work to do that. We need to, like I said, we need to sort of build an intuition around these things, and yeah, hopefully taking these measurements helps us get there.
Okay, so I'm going to segue slightly in terms of, you mentioned Chat GPT and intuition. I mean, we're moving at some pace now in terms of new technologies entering this field and entering this space. How disruptive do you think they're going to be, and what would be your thoughts in the space?
So I don't know. I mean, it's very interesting. It's a very interesting field. I mean, especially basically the idea is that, typically in a small molecule drug project, you find a protein that's causing a problem. You find something that binds to it, and here we're just saying, okay, well, let's just downregulate that protein from the get-go. So you have the genetics. You can type out the code, and in terms of finding something that binds, I mean, RNA and DNA, I mean, that's what they do. So it's not so problematic there. But yeah, I guess I could see it being very disruptive, actually. Or I mean, maybe what's the opposite of disruptive? Very helpful. But yeah, no, I think it really will change. It will change sort of, how we think about finding disease targets and how we treat them. And yeah, I mean, it really does fit into that kind of big data sort of language models to come up with these types of things. So yeah, I think it's very exciting. But I mean, I still think, we need to figure out just that biophysical problem. How do we get to that right cell? Because once you solve that problem, then you can just type up what you want and get it to where it needs to be.
Amazing. Okay, is that a view that's shared widely? I don't know. I wonder. I hear different scientists saying different things.
I don't know.
If it's a common view or if it's just you.
I think this view is this is probably more of a computer scientist's view, actually.
Okay.
people who are doing the biology, I mean, they know that these problems are very hard. So
all right. Okay, so moving on now to some of our questions which have come in from the audience. We have a technical question, I think, to start with. Okay. Why did you decide on a 34S rather than another stable isotope label?
Yeah, that's a really good technical question. So I can tell you that maybe this didn't come across, but you add a label and you can increase your sensitivity by adding more labels. And so we started out with carbon 13, and we got like pretty good sensitivity. And so presumably there's lots of carbons. You could add maybe, I don't know, let's say like 100 carbons to an oligonucleotide to increase the sensitivity. But it's not super practical. And so the reason the C13 needs so many labels, or the reason the sensitivity is bad, is because the concentration of carbon in the biomass and the epoxy, it's something like it's 600 millimolar. It's very, very high. And so you need to have like 1% higher than that to make a signal. The 34S is a much lower concentration in the cell. So to get above the background with 34S is easier. I mean, there's less places to attach a label. I mean, there's not as many sulfurs as there are carbons, but because the how would you say, the endogenous level of sulfur 34, which is coming from the natural ratio of sulfur to sulfur 34, is lower, you can increase it a bit. You can see the signal easier than with the carbon 13.
So that makes sense. Thank you for clarifying.
Sorry, a little bit too, yeah, but
No, it makes sense. It makes sense. Thank you. Our next couple of questions are about the different sections of the cell, different organelles within the cell. And the question that I first want to ask you is about the therapeutic index of any particular drug and whether or not it differs in different organelles in the cells to which it's targeted, and whether or not mass spectrometry can address these like minute differences between
yeah them. I mean, that's quite interesting, actually. I mean, so I guess the idea of a therapeutic index is sort of the concentration you need to fit into, and if it's too high, it's bad for you, and if it's too low, it doesn't give the effect. I would say, I don't know, at the subcellular level, I mean, it's not, at least for the oligos. We know that like drugs like amiodarone, for example, so these drugs will build up in the lysosomes and cause phospholipidosis. The cells become foamy. They have a lot of endosomes, and they're sort of degrading membranes. So that would be an instance where you could look at a subcellular therapeutic index, where if the drug is trapped at a certain level, it's okay, and then at this other higher level, it would be toxic. In terms of the oligos, I mean, we would have to take those measurements and see where those concentrations fit in. If I understand the question. So yeah. I guess like if there was a concentration related toxicity in if there's a concentration level related toxicity in the cell, which was related to the amount that was concentrated in the endosome, then yes, we could at least take the measurement. Whether or not if the margin is very small, then of course it's just a tough measurement.
Okay. Thank you very much.
Yeah, sorry.
Our next question is kind of related, but it asks if you've thought about trafficking these drugs within the. Sorry, I'm going to read the whole question out. You treat the endosomal space as one compartment. Have you thought about trafficking these drugs and whether you can pinpoint the exact structures that are responsible for the release of the drug into the cytosol?
Yeah, I mean, that's kind of like the that would be the real goal. I mean, if we could track down exactly where the escape would happen, then you would have a structure that you would want to target with some other type of treatment, or maybe even if you could manipulate the timing or get the drug to stay in the leaky structure longer. And we have been thinking about ways to do that, but right now our sort of philosophy around this is that that escape is we think that the concentration is related to that escape. So the more you have inside the endosome, presumably that helps more will get out. So if we think about like a model of this, so if the escape happens due to some defect in the membrane, right? And so you can imagine that when the endosome is forming, there's many fusion events that happen that bring the lipid in to make the endosome. And so if every one of those fusion events causes a defect, like a pore, so if the concentration is high inside and the concentration is very low in the cytosol, you'll create a diffusion gradient. And so the idea is that if it is about these defects, the higher concentration will give you a higher probability of the molecule to escape. And if you have another model where you say, okay, the escape is more of a stochastic rupturing. So every hour, one of these endosomes bursts. That's also related to the concentration, because the higher concentrations, the higher concentration endosome will release more molecule into the cytosol, and thus it's still related to the escape. So that's sort of how we're sort of taking we're working our way there. But yeah, we definitely do want to think about orthogonal methods to label specific organelles and see what those effects are.
Okay, great, thank you very much. A quick reminder to our live audience, you do still have time to ask a question. To do so, please type it in where it says type your questions here, and then press submit. Okay, so moving on then. The next question asks, what would you recommend to test the drug concentration using your linker strategy in macrophages? Are there any alternatives to tags?
So I guess alternatives to tags?
Yeah, to tagging. So it's asking what you would recommend to test the drug concentration in your linker strategy. So rather than tagging it, do you have any other alternatives that you have used or could recommend?
Well, I mean, I guess we don't really tag. I mean, we have labels incorporated into the starting material of the molecules that we made. I mean, depending on the molecule, I mean, I would say if you can find something that's halogenated, that would be the best step. I mean, I mean, I guess macrophages are sort of these. They're taking big amounts of volume into the cell. I mean, I would make sure that the molecule was fixable, of course. Otherwise I guess I don't really have. I guess I would need more information to help with the suggestion.
Okay, so that's potentially something we can follow up on after
the
finishes. Great. All right. Our next question asks if it's possible to compare this method to in vivo microdialysis, and if you think there are any advantages over that.
In vivo microdialysis. Yeah, I mean, I guess I'd have to think about it, because I mean, when you talk about in vivo microdialysis, you're talking about injecting a dialysis cannula and measuring plasma over time, I guess.
I guess so.
And I mean, so then you would get this. But still, I mean, if that's more of a real-time measurement, I mean, the SIMS is definitely more of a static measurement. So you would have to figure out how to get to sample at different time steps. But I mean, my feeling is that, you know, this is just a piece of the puzzle. And so whether it's the efficacy of the drug or the plasma concentration or the tissue concentration, it's really just another piece of the puzzle. So I mean, I would combine it with everything, I guess.
Yeah, combining things can often be revealing, right? So one and the other over there could be useful. All right, a reminder to our live audience, you do still have time to ask a question. To do so, please just type it in. Another technical question for you. Did you try high pressure freezing or freeze substitution? And do you think that would have any effect on drug concentration?
Yeah, that's a very interesting question. We did do high pressure freezing and freeze substitution, and we assumed. So I think the assumption here with the high pressure freezing and the freeze substitution is that it is a better way to prepare the samples, or at least for drugs, because you freeze the material really quickly, and so that the drugs are trapped really well, and then you raise the temperature a bit, and then you infuse the embedding media in at a low temperature, and so this should reduce diffusion. In our experience, it seems like it's really more important that the molecule is fixable. So for the oligos, they have a lot of arylamines, which can take part in this fixation process. So glutaraldehyde, formaldehyde, they make this bind. So there's lots of places for it to be grabbed on, and so we didn't see a really big difference between the two. And I think that at least in our hands, it seems like the fixability of the molecule is really important.
Okay, thank you very much. Okay, the next question is really asking for your thoughts more than anything else. I mean, big pharmaceutical companies are famous for very high throughput analyses and for having compound libraries with millions of molecules. Could you comment on the throughput of this technique and whether or not it's a concern for you working in this environment?
Yeah. Oh, I'm very concerned. Petrified, really. No, I think I would say it like this. So if we go back to my pie in the sky type thing, we're gonna, chat GPT, how do I cure my thing? Yeah. It seems like the chemical space that we're working with could shrink a bit. And if we're only interested in certain sequences of similar molecules, so we may not need to do the kinds of screening that are typically needed. Also, I don't really see this as screening I don't foresee this being something that you would do every experiment. I really think of it as like a fundamental type screen where you sort of figure out the margins, the places where you need to be in terms of to get effect, and then sort of if you're going forward with a molecule and the theory doesn't hold, so a drug designer maybe designs a drug to reach a certain cell in a tissue, for example, and so they've designed it based on exposure. They want to get the drug to the place, and so right now what we do is we measure the effect. And if there's no effect, you get to this place where you're like, okay, well, I mean, we designed it to get there, but how do we know if it got there? And or did we take the potency down by changing it to get there? So it's just sort of like, okay, we have a something that breaks a rule. Let's figure out if we did what we did. Did we actually design it to get there? And if it did, then you can start thinking about other things that may be affecting what's going on downstream.
Thank you very much. I think probably our last question for today is related to a sort of a point that you mentioned earlier, where you sort of said or mentioned that you need to give scientists working with pharmacokinetics data that they can understand. So can you elaborate on what that means in the context of what they do in a drug company?
Yeah, yeah. I think if you think about what the drug company does is, I mean, they really they collect data and they make decisions, and the decisions that they make, they're always trying to push the drug molecule forward. So how do we make the most useful therapeutic. And part of that, of course, is this data-driven approach, but also part of it, and I would argue a big part of it, is this kind of working model, this kind of intuition about how things work from hundreds of years of drug discovery. So it's you get this data and you've had these experiences and you've learned these things and what this data means, and that allows you to make decisions based on your experience. And I think the thing with this, the NanoSIMS measurements, the measurements of drugs and the endosomes, there is no working model. There is no intuition built up by it, because we have just only started measuring it. So I think that's a key point. But so for me, I thought that it would be a step closer if I could provide a concentration, because everything else that they work with is in concentration. So I thought that, yeah, that would be sort of a language that they could understand a bit better. And I have to say, I mean, it has been very well received, and it started a lot of conversations, right? So we're starting to talk about these things in that way. And I think even in my few years at AstraZeneca, I think at first we talked about uptake and endosomal escape and trapping, and they were all kind of these concepts. And the longer I've been there, and it's not just me, obviously, it's the whole landscape changing, but people do talk about it a bit different, and they do think of it in sort of in the details of it. And so I think I think it's changing, and I think it's an important measurement, and I think it fits into what we do really well.
And where do you see this going in the next few years?
I mean, I would like really would be, I'd like to sort of take more measurements, see, and with an eye on what potency is, like what concentration do you need to have an effect, what concentration is potentially bad, and then sort of see if we can link what we see in vitro to in vivo. So right now it's very difficult to make those connections, and obviously we want to be able to. It would be better to be able to screen in vitro than in vivo, just because of the you understand.
Risks. Yeah. Because of the risks.
Right.
Okay. Well, I'm going to thank you very much for your time today, for your presentation, and for answering the audience questions. I would also like to thank our webcast sponsor, Kameka, and of course you, the audience, for tuning in to this webcast today. Remember, you can watch this webcast again at any time on demand at nature.com/webcasts. Thanks very much to you, Michael.
Thanks.
Thanks for watching, and I hope you'll join us again soon.